My name is Adriano Ortiz, and I'm going to talk about my research experience in my laboratory. The title of my project is CRISPR transcriptional expression in Mycobacterium tuberculosis of candidate genes in pyrazinamide resistance. The Bioinformatics and Molecular Biology study group of the University Peruano Cayetano Heredia performed RNA-seq in Mycobacterium tuberculosis strains sensitive and resistant to PCA to evaluate the effect of the drug at acid pH. In this study, two induced genes were identified, RB3854 and RB2661, as a response to pyrazinamide tolerance. Therefore, this study aims to evaluate the effect of transcriptional repressions of these genes on PCA resistance. Thanks to this information, we expect to confirm the importance of these genes in the mechanism of action of the drug. What are the objectives of my project? Well, the objectives are to generate strains of Mycobacterium tuberculosis with loss of function in these genes using CRISPR interference technology. And the second is determine the PCA resistance of Mycobacterium tuberculosis strains with silenced genes. What are the biggest challenges and how did I solve them? Well, one of my biggest challenges was to identify the mistakes that prevent silencing. The first drawback was not detecting the absence of one adenine in the guide RNA after the cloning process. The cloned plasmid with the adenine deletion transformed it into Mycobacterium tuberculosis. And after inducing the silencing with tetracycline and idris, I obtained a repression fall of 4. During that time, I optimized the growth time of my bacteria before induction and also improved the quality of the extracted RNA, thinking that it could improve my silencing, but I only got a repression fall of up to 15, being 216 the reported repression fall. Therefore, I analyzed all my results, finding that I had working with a plasmid with 4 adenine. Possibly the absence of one adenine generate the inadequate formation of the guide RNA loop that could interfere with the binding of the Cas9, preventing the repression of the gene of interest. Then I cloned again with the same reagent but only obtained plasmid cloned with 4 adenines. So I decided to clone with the same reagent reported in the article, and I obtained plasmid with 5 and 4 adenine. So after reading more about these enzymes, I decreased the incubation time of my plasmid with the enzyme and obtained in all of my clones 5 adenines. Optimizing the cloning protocol. The second challenge was to decrease the standard deviation of the silencing repeats, since the one reported was 10, and in my essays I obtained one of 30. After analyzing my reagents, I noticed that the drawback was the DNA enzyme I was working with, because the percentage of DNA degradation was not optimal for my assay. So after changing the enzyme to a better brand, I decreased the standard deviation. However, I also decided to increase the volume of ATC dissolved in ethanol, because when working with a small initial volume of 5 microliters, it evaporated, and at the end I added a higher and variable concentration of ATC, causing the silence values to be very dispersed. The third challenge was the standardization of the CRISPR interference technique in Mycobacterium tuberculosis, because it is a slow-growing bacteria. Therefore, all of my work was meticulously scheduled, and after each opportunity for improvement, possible additional standardization were scheduled. This way, I made the most of the time, but advancing experiment at the same time and starting trials prior to the result of my previous one to reduce the waiting time of the growth of my bacteria. What are the most important things I learned? Well, first, the organization of my experiments is very important, specifically planning for possible delays or additional repetition. Also, optimizing time to obtain fast results, but respecting the analyzing time in case of unfavorable results. Second, to keep a record of absolutely all hits and misses of my experiment. Third, one should completely analyze the advantage and disadvantage of the reagent one will be working with. Third, maintain frequent communication with your advisor regarding the progress of your experiment to receive different points of view and identify opportunity for improvement.